Hello, my name's Alex Forrest-Hay and I represent Metabolon. Unfortunately, I didn't get to give this presentation at the meeting, so thanks for tuning in. And we've called this presentation A Little Less Association, A Little More Action. And it's really going to focus on the use of metabolomics as a route to understanding causality. And in this presentation, I'm going to talk a little bit about Metabolon, give some examples of how metabolomics has been used to empower more understanding of genomics, and a couple of examples about how it's been used in publications to identify causal metabolites linked to disease, and then just a little bit about Metabolon and how we generate the high quality data to understand metabolism. So just a very quick start. Metabolon is the world's leading health technology company that's focused solely on metabolomics. And we've spent the last 20 years, 19 years of our existence really building the competency around generating the highest quality metabolomics data that we possibly can and understanding the technology around metabolomics and interpreting the data. And we're now using this technology to power precision medicine and life science research. We're a privately held company. Our main lab is in North Carolina. It's a very large facility that now has the capability of processing 250,000 samples a year. But we also have labs in Europe and the Middle East and China. And our mission is pretty simple. We want to provide the most accurate, unparalleled, actionable knowledge of the phenotype using metabolomics to prevent, diagnose, and treat disease quickly and accurately. And we've done a lot of work over the last 5 years to really make the technology robust enough to run on individuals and make informed healthcare decisions. So in a nutshell, we have unrivaled capability to deliver the highest quality data, and we call it precision metabolomics. What we've done over the last 10 years is build out the world's largest library of metabolites. So think about this of, we actually have about 5,200 plus named metabolites in our library. These are biochemicals that we have seen in our studies. We have matched standards for them, and we know exactly what they do and what pathways they reside in and what function they have. And then we have another 2,000 metabolites where we've seen them reproducibly in many studies, but there isn't yet a known standard for those metabolites. So we call those unnamed metabolites, and we have over 2,000 of those. And every year, we have a rigorous R&D effort to try and understand what these new metabolites are and what their consequences are for biology. So it's really an obsession of the company to try and understand how you take inherently messy and noisy mass spec data and turn it into biological insights. And we've now done over 10,000 projects, many different types of sample. The most common is plasma, urine, fecal material, but we've done all kinds of different samples, including dog and apparently dinosaur. And we've studied over 500 diseases, 800,000 biological samples, or I believe that's actually higher now. And we're very prolific publishers of high impact journal papers. So in 2018 alone, we did 250 publications, and 27 of those were in high impact journals, Nature, Science, Cell, PNAS, etc. Overall, we have over 2,000 publications in this space. So we're obsessed by being as good as we can be at metabolomics, creating the best and most reproducible data, and more importantly, being able to interpret that data and taking it into meaningful biological studies and making important discoveries and interpretations from the data. So there are many applications for metabolomics. I mean, it can really address some of the most difficult and challenging needs in life science research. And as you look around this continuum here, you've got wellness. So we want to understand wellness, and we want to understand how patients transition from being essentially healthy to having a disease. And of course, your biochemistry in your blood gives you enormous insights into this, and you can actually see very early on symptoms of disease, or rather not symptoms, but actually biochemical changes in your profile that actually show that you're on a trajectory to a disease. So in this world of polygenic risk scores and understanding certain individuals or groups of individuals may have a higher risk of disease, being able to have a technology like metabolomics that can actually point you to those individuals that are developing the disease very early on is going to have a huge impact for healthcare. And equally, when you look at biomarkers or surrogate markers for safety and efficacy in drug trials, metabolomics helps you identify asymptomatic patients, helps you intervene early, and to adhere to these, to make sure you adhere to these drug regimes. Because not only can we discover the metabolic changes that happen when you take drugs, but you can actually monitor the drugs themselves. So a very powerful technology for that. We're able to predict disease and outcome, and that helps us in therapy selection and prognosis for patients. More and more interesting data is coming out using metabolomics as a functional readout for the microbiome. So understanding bacterial flora to improve therapy selection, drug response, and identify clinical pathways is becoming a really, really hot topic. And really, if you're doing 16S sequencing or metagenomic sequencing, you really have to have a metabolomic profile alongside to understand what the changes you're seeing in the gut microbiome, what impact they're having on the host. What I'm going to talk a lot about today is how metabolomics can be used to understand the genome, bridge insights across large population cohorts, find gene function information, match SNPs to causal outcomes. Very important part of metabolomics, and it's a really powerful part of what we do. And that also includes linking the genome to the phenome. So, Metabolon or the metabolome is the closest thing to the actual phenome. So you can really understand disease risk right through to manifestation of that disease, and also patient stratification. So enroll the right patients in the right drug trial and improve efficiency. And we heard in the conference a lot about drug trials failing at a late stage due to lack of efficacy. And really the ones that we think are most successful are the ones that combine genomics and metabolomics to identify potential markers and then understand what impact those markers are having on the patients who take those drugs. Understanding basic biology, understanding metabolic pathways and pathophysiology. Biochemistry has been around for hundreds of years and we understand it very well. We have a great ontology and an understanding of biochemistry. So studying metabolomics helps you really understand metabolic pathways and how they're changing when diseases manifest themselves. And finally, drug development is a huge area where metabolomics is having an impact. It helps you choose the right target, it helps you find the right biomarkers, and it helps you pick the right patients. So we have a long-winded way of saying metabolomics has a very, very broad application. And when it comes to Mendelian randomization and understanding causality, it's really helping understand gene function and linking the genome to the outcome where metabolomics is most powerful. So the first seminal paper that came out looking at metabolomics within a GWAS study was published in Nature in 2011. And this publication was done with Professor Tim Spector, looking at the Twins UK cohort and also the Cora cohort from Germany. The rationale here was to use a global metabolomic platform from Metabolon to phenotype two large genomic studies and to try and understand the connection between the genetics and the metabolome. And so this is a very deeply phenotyped cohort, two cohorts. They have genetic data, they have environmental data, they have SNPs, obviously, and they have clinical phenotypes. So we're able to really explore very deeply the associations between the genome and the metabolome. This was done using a smaller version of our platform back in 2010, which measured about 556 blood biochemicals. But what we saw was quite remarkable. We saw 38 genome-wide significant associations to biochemicals in the discovery study. These had never been reported before, and they were also completely replicated, or 37 were fully replicated in the second study, which was quite remarkable because these two studies were quite different in terms of the way that they were set up. But they still, all of these associations pretty much replicated. And what we saw underneath that data was some very exciting data that didn't reach statistical significance, but suggested that we could actually map the function of many, many more genes by increasing the sample size. So we did this in 2014 in Nature Genetics, a publication, Shin et al. Publication called An Atlas of Genetic Influences on Human Blood Metabolites was published. And this was done in collaboration with Pfizer, who enabled us to expand on the number of subjects we looked at in these two cohorts. And by doing that, by looking at 8,000 subjects and combining metabolomics with genotyping, we're able to explore 20 new gene functions. From a relatively small sample size, we're now really understanding novel gene functions in a very impactful way. And so this really gave us the ability to look at how a gene locus affects a metabolic trait. And we found 145 metabolic loci with associations, and 84 of those were completely novel, never been seen before. So very exciting data. And then there was a publication in 2017 by Long et al., which went into deeper insights into the genome by using whole genome sequencing, and looked at almost 2,000 twins from the UK twins cohort at 3 time points. So this gave us the ability to actually look then at how the metabolome is conserved over time, and whether we could see what the heritability is of your metabolome, and tracing it back to the genetics of these twins across 18 years. And by using whole genome sequencing, this enabled us to actually map 246 metabolite associations to common and also low frequency variants. And again, 90 of these associations and these were completely novel. So really impressive studies. And it shows that by adding metabolomics to GWAS studies, this reveals gene metabolite associations that can lead to defining new gene functions. And obviously that can help empower better Mendelian randomization studies. And so just looking at that Shin et al. Paper with a little bit more of a fine analysis, we actually analysed within that paper the medical and pharmacological relevance of these metabolic associations. And it was really interesting. If you look at the ones in bold, they are the ones that were genes of known function, and the ones that were not bold were the ones of unknown function. And so here we saw 26 genes associated with inborn errors of metabolism, and these gave us the potential to look at new biomarkers. We've got 10 genes that are targets of 45 drugs, and the rest were targets of around 400 drugs that were in development. We have 36 genes linked to complex disease traits and drug responses, and then we have involvement in other aspects of human health and disease, many associations of different enzymes and transporters, etc. So really powerful knowledge base that was created from this study of 8,000 samples. Going beyond that, when you look at the Long et al. Paper, the whole genome sequencing identifies common rare variants associated with human blood metabolites. This was more interesting because we were able to do it longitudinally over time, and this allowed us to really map gene variants to metabolite levels. And so what we actually saw here was very interesting. So we saw that 644 metabolites have consistent heritability over 18 years and across the twins. So basically what we're saying here is that we detected in this study just over 1,100 metabolites, I believe. So about 2/3 of those metabolites are controlled by your genetics. So they are essentially maintained over your lifespan and controlled very strongly by your genetic makeup. We also saw that 246 metabolic associations mapped to these common and low frequency variants and identified 90 novel findings. One particular finding that was interesting is an amino acid called dimethylglycine, which is actually linked to an inborn error of metabolism. And it's linked to DMGDH, the gene, and the GWAS loci. And so what we saw here was that over time, if you can actually see here, we see that the levels of dimethylglycine are fairly well conserved over 18 years. So this is time visit 1, 2, and 3, separated by 18 years. But there were 3 sets of twins here that consistently had higher levels of dimethylglycine over that period of time. So again, controlled, but very much higher. And we actually identified with this study carrier mutations and loss of function variants that actually led to the higher levels of dimethylglycine in these twins. And this is really important because now you can see that you can actually, even though these twins were asymptomatic, there's no disease associated with this, you can see biochemical changes that are being caused by specific gene variants. And just a quick peek, Adam Butterworth and Claudia Langenberg and their teams are working on an even larger set of GWAS now with Interval and EPIC Norfolk, and they are working on a publication that really is now starting to map out many more genetic regions that have metabolic impact. And so with this paper that's going to come out, what was this data that was presented at ASHG last year showed that 2,647 independent variant metabolite associations were seen within 330 genetic regions. So really combining genomics with metabolomics really enables you to understand the genome much, much better and see these links between genotype and metabotype. So I want to give an example now, we've got a few minutes left, an example of how serum metabolite can be found to be causal for increased blood pressure. This was a really nice study in hypertension by Valdez et al. And basically it showed very clearly that hexadecanoate was highly associated with blood pressure in 3 independent cohorts. And by using metabolomics, they were able to use deep metabolomic profiling to actually functionally map the SLC01B1 gene, transporter gene, and the SNPs within that gene to hexa. And so from here, they did a separate in vivo study using mice where they actually injected them with hexa and saw that they had systemically higher levels or higher blood pressure levels. So really just showing the causal role for hexa in high blood pressure. And then from there was a further study done in the ARIC study by Eric Bowinkle's group, where they thoroughly explored the loss of function variants within SLC01B1 and showed that, that certain variants increased hexa levels, and indeed it was dose dependent. So one copy of the mutated allele increased risk of heart failure by 29%. So this is a really beautiful study to show how metabolomics coupled with genomics really helps understand the power of marrying metabolomics with genomics. And this is important because by 2020, over 1.5 billion people are predicted that they will have high blood pressure, and 2/3 of them will not achieve target therapeutic control with current therapeutics. So there's a huge opportunity here to use this data to potentially develop a drug. And so what we're seeing is a huge increase in population health organizations and cohorts investing in the power of metabolomics. You've seen some data from Twins UK and provisional data from EPIC, but there's now over 70 cohorts that have started to run thousands of samples using Metabolon's technology, including ones here from Bristol. We have a great relationship with Debbie and with Nick, doing some really nice work in HealthPack and BIB. And in the US, we're working with the Million Veterans Program. And we've recently signed a contract with the Canadian Longitudinal Study of Aging to look longitudinally at diseases associated with aging in a cohort in Canada. So some really exciting things coming along, and there's going to be a lot of data for you guys to play with in the coming years as these programs start to expand. And so just want to close out by just saying, how does Metabolon do this, and what makes us very special in this space is that when a sample comes into Metabolon, into our lab, we essentially perform biochemical extraction on that sample, and then we run it through multiple methods. So we have the top 4 methods here are what we call our global metabolomics platform, and that takes about 100 microliters of sample, splits it up into 25 microliter aliquots, and runs them through these different chromatographic methods to capture as many metabolites as we possibly can. And then we also have a separate method to look at complex lipids. And together this allows us to actually profile over 2,500 biochemicals that are maintained homeostatically in plasma in a highly robust manner, in a fully automated manner. So we call this precision metabolomics, but that's really just generating the data in a highly robust way with high levels of stringency. From there, we actually have a bespoke and very complex and impressive piece of software called the metabolizer software, which takes that library of metabolites that I talked to you about earlier, of about 7,000 biochemicals, and it takes the peaks and the area under the peaks and screens them to give us a biochemical readout along with relative concentrations of each of the biochemicals seen. And you can also see in there, as well as obviously tryptophan and cholesterol and all the usual things that you would expect to see, there are also X compounds in there, which are highly reproducibly identified. And these are the unnamed compounds that I was talking about earlier. So what we're able to do in an automated way, in a very fast way, and an accurate way, is actually take you through the noise of the data that comes off the mass spec system and the chromatography columns, and using retention time, master charge, and MSMS spectra, match those to known metabolites. So very, very powerful technology, which enables us to give more metabolites, more accurate, and and the broadest coverage that you can get, which is very important when you're doing discovery work. And just to go further into that, we really cover every known pathway and we've actually mapped out the metabolic map in humans. And so, as well as capturing endogenous metabolism and biochemistry, we of course also capture microbiome metabolism, drugs and antibiotics, drug metabolites, byproducts of host metabolism, as well as all of the complex lipids, etc. So really covering as much of the biochemical spectrum as you possibly can with one sample. And talking about the microbiome, we have identified about 200 molecules that we can identify robustly in our platform right now that we know are derived from bacterial metabolism. So here you're getting genetics of the host and the biochemistry of the host. You're getting external influences from the environment, and you're also seeing the functional readout of the microbiome. So overall, you're really getting a very nice view of what's actually happening within that patient at the time the sample was taken. So real health readout. And this is really exciting. The blue, blue chemicals here, biochemicals here, are exclusively or mainly contributed by bacteria, and the red ones here are contributed by mammalian cells and bacteria together. And so, a couple of things just to emphasize before I close. We have over the last 2 years developed the ability to compare data sets across cohorts. This is absolutely critical because there's been issues with batch effects affecting metabolomics data sets for years, and it's been one thing that stopped the field from really expanding. But now, because of the consistency that we generate data, we now have bridging strategies that enable merging across large data sets, and that's exactly what's been done by Adam and Claudia's group to combine data sets across multiple cohorts. And so by using a standardized matrix control for all samples, this enables matrix normalization to be used, and data sets can be merged with minimal batch effects. And we can now automate the curation of the data, which enables us to update this on an annual basis. So if you think about this in terms of next generation sequencing, we're collecting all the data from any given sample, most of which we understand what it is, but some of which we don't. And over time, using this automated curation and our quest for understanding what all of these unnamed molecules are, we can update that data from annually to make sure that you have the latest information. And because we have so many samples now being run through our North Carolina lab, we have the ability now to run very large projects very quickly. And that's a very, very important development as well, because we want to be able to run tens or hundreds of thousands of samples, potentially do something like UK Biobank, but do it in a reasonable time frame. And that's certainly a goal for metabolomics moving forward. So I'm just going to conclude by saying, fundamentally, allelic differences in individuals cause metabolic variations in subjects, right? So we've seen that, we've linked the genome to these metabolic variations, and that's a really important surrogate phenotype and a really important way of understanding gene function. I didn't touch on it too much here, but MQTLs point at the causal gene or the causal pathway, and a lot of the biochemical interrogation has already been done pre-GWAS because we understand the ontology of biochemistry very well. So MQTLs are really a very powerful way to understand causality. And we can use this approach to define function for potentially any gene that will have a metabolic impact. And so that, as we ramp up the number of samples, we're going to understand gene function so much better by incorporating metabolomics with genetic studies. And metabolomics can uncover fundamental relationships between disease and metabolism. And the Metabolon technology is highly scalable, and it's precise enough to do across large cohort studies. So thank you very much for listening to this, and appreciate it. If you have any questions, I will send my email address alongside this presentation, and I look forward to hearing from you. Thank you very much.